Hello students, welcome back to my channel. Today I am starting a new video on the absorption by roots. This is the second part of this topic and this topic is from class 10 Icse. Now if you haven't watched the first part yet, then first go and watch that part in my channel and then come back to this video for better understanding. So let's start the video. Now first is, what is osmosis? Now, osmosis is basically the movement of the water molecules from their region of higher concentration to their region of lower concentration. So, I have already told you about diffusion, but in osmosis, what will happen? Osmosis will depend upon, or it is basically all about the movement of water molecules. Only it is concerned with water molecules. Here in the process of osmosis, the water molecules move from where to where? From their region of higher concentration to their region of lower concentration. That means what? Water molecules will move from a region where water molecules will be present at higher amount to a region where water molecules are present at lower amount. So see, in this picture, which I have already provided you in the screen, this is actually the solute particles, and these yellow structures, these are water particles. See in which side the water molecules are present more, left or right? Obviously in left side the water molecules are present more. So look at the arrows. The water molecules are moving from left to right because the region of higher concentration of water molecules is at left to the region of lower concentration of water molecules, which is at right. And another thing which is very important, this process, that means osmosis, this movement of water molecules will occur or will take place through a semi-permeable membrane. There is a membrane which you can see, semi-permeable membrane. So what do you mean by semi-permeable membrane? Semi-permeable membrane is a membrane which only allow the water molecule or the solvent to pass through. It will allow only the water molecules to pass through it, and it will not allow any solute to pass through it. Okay, only it will allow the water molecules. So this is known as the semi-permeable membrane. So again I am repeating, osmosis is all basically depends upon the water molecules, the movement of the water molecules. And where will this movement of water molecules will occur? The movement of water molecule will be from the region of their higher concentration, where water molecules are present more. Like in this picture, in left the water molecules are present more, and they will move towards the region where water molecules are present less. So from left to right the water molecules will move. The movement will be from left to right. And another important thing in osmosis is it will occur through semi-permeable membrane. So semi-permeable membrane is a membrane which allows the passage of the molecule selectively. It allows only the solvent to pass through it freely. Like the water molecules, it will allow to pass through it freely, but it will prevent the movement of the solute particles like sugar molecules, salt molecules in the solution. Okay, it will not allow. It will only allow the water molecules to pass through it. Now we will study about the next two terms, next two types of osmosis. One is the endosmosis, another is the exosmosis. So first that I have provided with example. So let's look at this. First one is, see, the resins. If you dip few resins inside a bowl containing water, what will happen? After some time you will see that those resins have swollen up. See, they are looking like grapes because water has entered inside these resins. Why? Basically what is happening, when you have kept these resins inside the beaker containing water, then water is present more in the beaker or the bowl. It is present more in the bowl rather inside the resin. So The water is present where? More it is present in the bowl than the resin. So the water molecules will, what will they do? They will move from their region of higher concentration to their region of lower concentration, as water molecules are present more in the bowl. So they will move more inside these resins. Inside these resins they will move more and more. So water molecules will be moving inside the resins, and these resins will they will swell up. This is known as endosmosis. Endo means inside or inward. So in the picture which I have provided in the left, in this what is happening, again I am repeating, if you take resins and if you dip those resins inside a bowl containing water, what will happen? The water concentration, the water molecules, obviously you can understand that the water molecules are present lesser in amount inside the resins, and the water molecules are present far more in amount in the bowl. So you already know that osmosis is the movement of water molecules from where to where? From the region where it is present more in amount to the region where it is present in lower in amount. So in this case it is present more in the bowl. So the water molecules will move from the bowl inside the resins. Okay, and as more and more water molecules will come or move inside the resin will obviously, they will swell up. So this inward movement of the water molecules is known as endosmosis. Endo means inward. Now if we see, if we take grapes, just the reverse. If we take grapes and if we dip that inside the bowl containing salt solution, okay, in this case it is salt solution. So in a salt solution, what will happen? The water molecules are lesser. Water molecules, the concentration of water molecule is less. Water molecules are present lesser than the solute or the salt. So in this case what will happen? Inside those grapes, the water is more in amount. The water molecules are more than the salt solution which is in the bowl. So they will come outside. The water will come outside. The water molecules, where it is present more? Water molecules are present more inside the grapes than the salt solution. So the water will come out because they will move from their higher region to their lower region. So in this case the water will come out. The grapes will shrunk. We will get shrunken grapes if we keep them inside the salt solution. This is known as exosmosis. Exo means outward. Here the water movement is occurring outside. Water molecules are coming out of the grapes. So this concept is very, very, very important. See, in the left hand side, where we have kept resins inside a bowl containing water, there the concentration of water molecules is greater in case of the bowl than the resins. That's why the water molecules are moving inside the resins, and so the resins are swelled up, right? But in case of the grapes, when we are keeping grapes inside a bowl containing salt solution means water molecules are less in that solution. So inside the grapes, water molecules are more. That's why water molecules are coming outside the grapes, because water molecules will move from a region of their higher concentration, which is inside the grapes, to a region of their lower concentration, which is the salt solution. Okay, so this concept is very important. So basically, endosmosis is, endo means inward, as I have told you. So it is the inward movement or diffusion of water through a semi-permeable membrane. Okay, and here what will happen? The cell will be kept in a solution which is less concentrated. See, in this case the solution is water, which is less concentrated. The concentration of solvent molecule, the concentration of the solvent is much greater than that of the solute. Such solution we will call it as less concentrated solution, right? And see, what will happen in endosmosis? The cell will swell up. And on the other hand, if you see exosmosis, exo means outward. So this is the outward movement or diffusion of the water, obviously through a semi-permeable membrane, but here the cell will be kept in more concentrated solution. So there will be a shrinkage of the cell. See, here in case of salt solution, the solute molecules or the solute particles, they are more in concentration than the solvent. Clear? So this is all about the exosmosis and endosmosis. I think it is absolutely clear to you all. So basically, what is the difference between the endosmosis and exosmosis? Endosmosis is the process that takes place when the cell is dipped in hypotonic solution or pure water. In my next video, I will tell you about the tonicity, hypotonic, hypertonic, isotonic. In the next part, I will tell you about this. So for now, just you remember that the endosmosis will occur when we will keep a cell in a pure water, and exosmosis will occur when we keep the cell in a concentrated, highly concentrated solution. In endosmosis, what happens? The water molecules enter inside the cell, right? And in exosmosis, the water molecules move outside the cell. So here the cell becomes turgid. Turgid means it gets swollen up. Turgid means it gets swollen up. And flaccid means it gets shrunk it gets, okay? Example is raisins. When you will dip in water, then endosmosis will occur. And grapes, when it is dipped in highly concentrated sugar solution, then what will happen? Then exosmosis will occur, right? So these are the basic differences between endosmosis and exosmosis. Basically, endosmosis means the inward movement of the water molecules. It will move inside the cell, generally when it is kept in pure water. And exosmosis means the outward movement of the water molecules. Obviously both will occur through a semi-permeable membrane, but exosmosis means when it will move outside the cell, the outward movement of the water. Generally when it is kept in a concentrated solution like a sugar solution or the salt solution, where the solvent molecules, the concentration of solvent molecules is less. Okay? Now let's talk about an experiment, this experiment is very, very important from ICSE perspective, because often this question comes in ICSE. So you need to understand this experiment very well. Okay. Now what is there? There is a beaker containing water. There is a thistle funnel containing sugar solution. Inside the thistle funnel there is sugar solution, and in the beaker there is water. Okay. So what will happen? And the mouth of the thistle funnel is covered with a cellophane paper. In this case, the cellophane paper acts as a semi-permeable membrane. The cellophane paper acts as a semi-permeable membrane. This you need to keep in your mind. Cellophane paper actually acts as a semi-permeable membrane. So what will happen? This is the initial level. If you keep this setup for at least 1 or 2 hours, then after that you will notice that there is a rise in the level. There is an increase in the level of the sugar solution inside the thistle funnel. This you will notice. Why this is happening? What do you think, student? Why this is happening? Because cellophane paper is a semi-permeable membrane, so it will allow the movement of water molecule. Now where the water molecule is present more, in water or in sugar solution? Obviously in water, the water molecule concentration is more than the sugar solution. So you already know that osmosis means it will occur through semi-permeable membrane. You already have a semi-permeable membrane in the form of cellophane paper, and there will be a movement of water molecules. But the water molecule will move from where to where? From a region of higher concentration to their region of lower concentration. So in the beaker, water molecules are present more. So they will move inside the thistle funnel, where the concentration of water molecule is less. As a result, the level of the sugar solution will increase inside the thistle funnel, because this cellophane paper is semi-permeable. It allows water molecules to pass through, but it will not allow sugar molecules to pass through. Okay? So that's why as the water molecule has gone in or it moves inside the thistle funnel, that's why there is an increase in the level of the sugar solution inside the thistle funnel. The sugar solution has not come outside the thistle funnel. As the water molecules are more in the beaker, that's why from the beaker the water is getting inside the thistle funnel. That's why there is an increase of the sugar solution. Got it? This experiment is very important. Now often question comes, what will happen if we use cellophane, if we use rubber sheet instead of cellophane paper? See, this I have provided a picture of rubber sheet. If you use rubber sheet in the same experiment instead of cellophane paper, then what will happen? Do you know? Rubber sheet is impermeable. It will not allow the passage of any molecule. It will not allow the water molecules from the beaker to cross over the other side. So there will be no increase in the level of sugar solution inside the thistle funnel, because no water will be able to move inside the thistle funnel. As rubber sheet is impermeable, it will not allow. There will be no change in the level of sugar solution inside the thistle funnel. Because impermeable means it will not allow any water molecules from the beaker to cross over to the other side. Okay. Now, if a question comes, what is muslin cloth? There is a slight mistake, typing error. So, what is a muslin cloth? Now, if you use a muslin cloth, what will happen instead of cellophane paper? Muslin cloth has pores. Okay, you can see that there are various pores in the muslin cloth. So, muslin cloth has pores, large pores. So, they will not be able to hold back any molecules in the solution. They will allow both the sugar molecules and the water molecules to pass through them, because it is freely permeable. It is freely, it will allow the entire sugar solution to flow through them. Okay. So what will happen? Not only the water molecules, but along with water molecules, the sugar molecules will also go inside. Or the thistle funnel. From the thistle funnel, the sugar molecules will come outside the beaker. So this will not give you the fruitful result. But if a question comes that if you use a whisking bag, this is a whisking bag. If you use a whisking bag instead of cellophane paper, what will happen? Yes, if you use a whisking bag instead of cellophane paper, then you will get exactly the same result. The osmosis will occur. Okay, because whisking bag is semi-permeable in nature. It will allow only the water molecules to pass through. It will prevent the movement of sugar molecules. So you will again see the same result, the increase in the level of sugar solution inside the thistle funnel. Okay. So only if you use the whisking bag, you will get the same result. Otherwise, if you use muslin cloth or rubber sheet, you will not get the desired result, because rubber sheet is impermeable, which will not allow any of the molecule to pass through them, whereas the muslin cloth has so large pores that it will also allow the sugar molecules to pass through them. So you will not get the desired result. So this question is very important. This experiment is very important. Often this question comes in Icse. So very, very, very important question. I think now the concept is clear. Now the question is, for how long can osmosis continue? Theoretically, osmosis should continue until a concentration of water molecules becomes equal on both sides of the membrane. Suppose these are the water molecules. Okay, and in red, these are the solute. So obviously this side the water is present more, so water will move from left to right. But for how long? Till that time when on both the sides the water molecules become equal. Okay, so in the experiment which I have already explained, the movement of the water molecules will continue until and unless in the beaker and in the thistle funnel both will have the equal concentration of water molecules. A stage will come, okay? A stage will be reached when no osmosis occurs, even if the concentration of water molecules is not the same on the two sides of the membrane. In this state, what happens? An equilibrium has reached. in between the beaker and the thistle funnel. There an equilibrium is reached till the concentration of water molecules become equal on both the side of the membrane. The osmosis process will continue, and that condition is known as isotonic. Iso means same. On both sides of the membrane, semi-permeable membrane, the water molecule or the concentration of the water molecule will become same. At that point of time, osmosis will occur. This is known as isotonic solution. Okay. Now, what is osmotic pressure? Osmotic pressure is the minimum pressure that must be exerted so as to prevent the passage of the pure solvent into the solution when two of them will be separated by a semi-permeable membrane. So, it is basically the minimum pressure which we will, or which the solution, or which the the solution is applying the minimum pressure. Why? So as to prevent the passage of the pure solvent into the solution. See, this is a solution. Here in the left, only pure solvent, the solvent molecule is present, and here the solvent concentration is less. So the water is moving from left to right. So osmotic pressure is that minimum pressure which will be exerted so as to prevent the passage of this pure solvent into the solution. And obviously there will be a semi-permeable membrane present between them. This you need to keep in mind. Okay, so this is the osmotic pressure. And the last, I would like to state the difference between the diffusion and osmosis. In my last video, I have already told you about diffusion. So now you will be able to know the difference between the diffusion and osmosis. See, the diffusion is what? The movement of all the three states of matter, the solids, liquids and gases. But osmosis is the movement of only the solvent molecules. So osmosis occurs only in the liquid medium, only in the watery medium. But diffusion can occur in all the three mediums, solid, liquid or gas, because it denotes the movement of solid, liquid or gas. In diffusion, there is no requirement of any membrane, but in osmosis, a semi-permeable membrane is required. Otherwise, osmosis will not occur. Right? And the last step is, or the point is, the diffusion process takes place from high diffusion pressure to low diffusion pressure. But osmosis process takes place from low osmotic pressure to high osmotic pressure. That means in diffusion, the diffusion pressure will take part, and in osmosis, the osmotic pressure will take part. So mainly the main difference is the diffusion is the movement of all the solid, liquids and gases, but osmosis is the movement of only the solvent molecules, only the water molecules and the solvent molecules. So diffusion occurs in solid, liquid or gaseous state, but osmosis will only occur in liquid medium. And another most important point is, in diffusion we don't require any membrane, but in osmosis a semi-permeable membrane is required, because without the semi-permeable membrane osmosis is not possible. Okay, so this much for today. I think I have made this topic absolutely clear. Still, if you find any problem or any doubts, you can comment in the comment section. I will definitely try to clear all your doubts. Okay, so please, if you haven't yet subscribed to my channel, please subscribe the channel. I need your support a lot. And please, if you think that my videos are informative and they're helpful, then please like and share my videos. If you share more and more, then people will know about my channel. And please support me. I'm a new YouTuber, so please support me and please subscribe to my channel. And stay tuned to my channel for more such videos. Thank you so much.